So today we're going to be talking about proteins, and we're going to be talking about what proteins are made out of, amino acids, how they're linked up together, how they have structure, and how that structure relates to their function. And we're going to be talking about the different kinds of reasons why we have proteins in our body, and end with an idea of how much protein should be in our diet and what kind of protein should be in our diet. Okay, so proteins in the body, these are incredibly important biomolecule, and as you can see from this diagram, that they are found virtually in every area of the body, and they're found doing every kind of activity, all the way from structural things like keratin in your hair, to energy production like the mitochondria in your pancreas, to building more protein, such as protein biosynthesis, all the way to making fats. So they're really they're a workhorse of the body. Okay, so what are proteins? Well, proteins are a polymer. They are a biopolymer, and they're a polymer just like any other small covalently bound polymer that we've been introduced to before. They are made out of monomers, and the monomers in this case are described as amino acids. And so we have this complement of amino acids. And they're called amino acids because each of these has two functional groups in it. They have an acid and they have an amine, so we call them an amino acid. And they're actually linked up through the amino acids. And so when many of these amino acids are linked up together, as you can see here, 1, 2, 3, 4, 5, 6. So we've got 6 amino acid monomers, and they're linked up into a chain. This chain, we're going to call this chain a protein. The protein is nothing more than a polymer of linked up amino acid monomers. And these polymers can get quite large. As the amino acids get linked up one by one by one by one, and you can kind of see them right here, right? So this is a picture of maybe 4 of these amino acids. This is these shorthand notation kind of tells us which of the amino acids, so cysteine, serine, leucine, phenylalanine, and each one of these once again has an amino group and an acid group, and they also can vary. So there's a side group that allows them to have some variation, and this variation causes them to twist and to turn and to have kind of a three-dimensional structure to them. And so they're actually these protein polymers take on these really interesting forms, and so these are the large scale views of these proteins, and these are 3 just kind of random proteins, and you can see that they do cool things. They corkscrew. They have kind of random coils, and they have these sheets kind of structures, these kind of flat sheets that will parallel each other, and then another kind of corkscrew, and so you end up kind of getting these kind of systems that kind of wrap around, and really it's this kind of structure, this kind of three-dimensional structure that gives these proteins its shape, or it gives these proteins its function. And so what happens if you unfold a protein? So you have this protein, which is this polymer of amino acids, and it's folded into this specific shape because of what type of amino acid sequence it has, and so you can unfold it, and you can unfold it in a way scientifically we call this denaturing. You can unfold it by heating it really hot. You can kind of shock it. You can chemically denature these molecules. So you can take a folded protein and you can unfold it. It's still, of course, the same sequence of amino acids as before. We haven't actually changed that, but what we've done is we've kind of changed how it looks, so it's not folded in the same way anymore. And a good example of this might be cooking an egg. So of course you have lots of these protein polymers in albumin. This is the white or the clear part of the egg, and then as you take these specifically folded proteins and you heat shock them, they ultimately will denature, and they will change their shape and form. They actually kind of harden, and you can see that as you cook an egg. You are denaturing the proteins. And the thing is that in many cases, when you denature a protein, it is unable to return to its natural state. So you don't spontaneously see the cooked egg go back to its original form. This is something that just doesn't happen. So you basically destroy the function of the protein by doing this. Okay, so if proteins are these polymers of amino acids and they have these three-dimensional shapes, so what do? Well, they do a lot of things. As I mentioned before, they're found in all parts of the body. One of the simplest things that they do is simply act as a structural unit. So muscle in the human body is a form of protein, actin and myosin. It's a way that our muscles kind of contract. Collagen is another great example. It's something which gives us form to our face and to our features. Keratin, as I mentioned before, is hair. It's going to be feathers on birds and horns as well. But the proteins can actually act more than just structurally. They can be they can serve very specific purposes. And a great example of this might be hemoglobin. Hemoglobin you can see is a protein structure. It's a very specific protein structure. And within the hemoglobin, it actually has these iron centers. It's got 4 of them. And each one of these can absorb an oxygen atom. And so hemoglobin is something that carries oxygen from the lungs throughout the body. So it basically distributes oxygen so that it can be utilized as a terminal oxidant and help kind of power some of the chemical reactions in your body. So a special kind of protein are enzymes. We've mentioned enzymes before in this class. So enzymes basically they induce chemical reactions in the body. So they basically make chemical reactions take place, and they're really quite important for allowing biochemistry to happen. And they take kind of a lock and a key approach. So you want to think about an enzyme as being kind of a lock that fits a certain molecule, which is in this case the key. And this molecule can fit in here, and when it's inside the enzyme can trigger the chemical reaction, whatever that might be. In this case, it might be breaking this in half into 2 separate molecules, and then it can release it as 2 separate molecules. Maybe a real-life example of this would be the enzyme that maybe would break down maltose. So maltose is a sugar molecule. It's a disaccharide. We're going to talk about this in a little bit, which basically means it's 2 sugar molecules that are attached to each other. And the maltose can get broken down by these amylase enzymes. So this fits inside the enzyme. The enzyme undergoes a chemical reaction, and you get out 2 glucose molecules, which are monosaccharides. So you're breaking this linked up sugar into individual sugar components that can be further metabolized by the body. 3 vital enzyme classes that we have in our body are proteases, lipases, and amylases. And so proteases, we have these in our digestive system. So when you eat a piece of meat, the first thing that it does is it actually takes all the protein in the meat, which are these polymers, and these proteases will actually break them back down into amino acids so that our body can take those amino acid building blocks and rebuild new proteins, right? So we can kind of use the amino acids for whatever we need them to use. Lipases are another one. So lipases are effectively help us break down or modify the fats so they can become utilized ultimately for either storage or for energy. And then amylases break down the sugars that we eat. Many sugars that we eat are starches, which are very complicated sugars. They're very complicated, complex ones that have to be broken down first into the smaller sugar pieces, and that's really what the amylases do. Okay, so finally, we should discuss about complete versus incomplete proteins, which is something that we hear quite a bit about. There are about 20 amino acids, and so these 20 amino acids are required for the construction of a whole bunch of different important protein polymers in our body. Our body actually can make 11 amino acids, alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, ornithine, proline, serine, tyrosine. So these ones we actually can biosynthesize ourselves. So even if we had a diet that was completely absent of these ones, we would still be fine because we actually can make them from some other pieces. But we have these amino acids which we consider essential, and these amino acids, these 9 right here, isoleucine, leucine, valine, histidine, lysine, methionine, phenylalanine, threonine, and tryptophan. These ones we must eat because we can't make them ourselves, and we need them. We need them. If we don't have them, we get very sick very quickly. If you eat meat, if you are a meat eater, you get all of these right off the bat. So meat is a complete protein. You get all the essential, and you even get some of the non-essential too. If you are into plants, if you're a vegetarian, then you have to be more cognizant of what you eat, and you need to be careful about mixing and matching because some plants are higher or lower in protein, and some are incomplete proteins. That is to say that they don't actually give you all of the necessary amino acids that you need to live. And so you have to be careful. There are a few a few plants that do give you complete protein, such as quinoa is a good example. But beyond that, you have to kind of make sure that you're getting the full complement for optimal health.